Good day, learners! I am Zarina A. Ruiz,
And I am Sarah Jane L. Dagdag.
and we are from 12 STEM B theories.
And now we're going to discuss about modifications to Mendel classic ratios.
Are you ready to listen? Then let's start.
Gregor Johann Mendel, an Australian botanist, is known as the father of modern genetics. He conducted an experiment with P, or known as Pisum sativum, in 1854 to 1856 primarily. Because of its numerous distinct varieties of traits, Mendel found out that every gene consists of two alleles. Remember that allele is a variant form of a gene. Moreover, alleles can be described as either dominant or recessive. Dominant alleles show their effects even if a gene is heterozygous or consists of both dominant or recessive allele. Unlike recessive alleles, which only show their effect if a gene is homozygous, i.e., two copies of the allele. But before we proceed to the main topics, let us learn first the difference between genotypes and phenotypes as well as the Punnett square.
According to Lewontin et al. 2017, stated that genotype is some relevant part of the DNA passed to the organism by its parents, while phenotype is the physical and behavioral traits of the organism. In simple terms, the genotype represents a genetic contribution to the phenotype. It is important to know the distinction between these two since they are closely related to the transmission of traits from generation to next generation.
Now, we use Punnett square to illustrate the probabilities of various combinations of alleles. It was invented by Reginald Punnett in 1905. It is a table where the genotypes of both parents are given to get all the possible outcomes of a genetic cross. As we can see, it is a square divided into 4 quadrants, containing one genotype per column and per row. This will be tackled further in later discussion.
And now, we shall move on to our main topic. Let's start with codominance. One of Gregor Mendel's contributions to the study of heredity was the concept of dominance. Mendel observed that the heterozygote offspring can show that same phenotype as the parent homozygote, so he concluded that there were some traits that dominated over the other inherited traits.
What is codominance? Codominance is a relationship between two versions of a gene. Codominance means that neither allele can mask the expressions of the other allele. It is a condition in which both alleles of a gene pair in a heterozygote or fully expressed, with neither one being dominant or recessive to the other.
Codominance is when an organism that has both alleles of a gene displays both phenotypes at the same time. Dominance affects the phenotype derived from an organism's genes. but it does not affect the way these genes are inherited. Complete dominance occurs when the heterozygote phenotype is indistinguishable from that of the homozygous parent. However, sometimes the heterozygote displays a phenotype that is an intermediate between the phenotypes of both homozygote parents, one of which is homozygous dominant and the other of which is in homozygous recessive.
When codominance occurs, codominance occurs when the phenotypes of both parents are simultaneously expressed in the same offspring organism. Indeed, codominance is the specific term of a system in which an allele from each homozygote parent combines in the offspring, and the offspring simultaneously demonstrates both phenotypes. Blood type AB is an example of codominance where alleles A and alleles B are both expressed. So, if an individual inherits alleles A from their mother and allele B from their father, they have blood type AB. In codominance, blood type AB implies that both dominant alleles are present and expressed together. Many blood proteins contribute to blood type, and the ABO protein system in particular defines which type of blood you can receive in a transfusion. In a hospital setting, attention to the blood proteins present in an individual's blood cells can make the difference between improving health and causing severe illness.
Black and white coat. In other animals, codominance is exemplified by a mix of coat colors in a progeny of parents with different coat colors. For example, a cross between a black-furred male dog and a white-furred female dog could produce offspring with a black and white coat. This means that the color coat traits of are codominant, as both were expressed together in the progeny.
White spotted red flower. Codominance is exemplified by a plant that bears flowers with two distinct color phenotypes. For instance, a white spotted red flower could be caused by a cross between a red flower and a white flower. The alleles for the red and white color phenotypes mean that they are codominant.
So, after discussing codominance, let's move on to our next topic, which is incomplete dominance. Incomplete dominance is a form of gene interaction in which both alleles of gene at a locus are partially expressed, often resulting in an intermediate or different phenotype. It is also known as partial dominance. For example, in roses, the allele for red color is dominant over the allele for white color. After Gregor Mendel discovered inheritance laws, the term incomplete dominance was proposed by the German botanist Karl Conners in 1864 to 1933. Karl Conners continued research and conducted an experiment on 4 o'clock flowers. This experiment leads to the discovery of incomplete dominance.
a condition in which a heterozygous individual doesn't show a dominant allele, rather show a phenotype intermediate of the phenotypes of the dominant and recessive alleles. To further understand the basic concept of incomplete dominance, some terms are defined briefly as follows. An allele is a form, version, or set of gene expressions. An organism consists of two alleles from each parent of one gene. The allele that masks or suppresses other alleles and becomes prominent in the offspring is called a dominant allele. The effect of an allele that is suppressed by the dominant allele and does not appear in an offspring is referred to as a recessive allele. Multiple alleles refer to the various alleles or two or more for the same gene. An organism that possesses two same alleles for a specific gene and can truly breed for the allele is described as homozygous. An organism that possesses two different alleles for a specific gene is described as heterozygous. A set of genes is an organism that is inherited by the offspring and that determines that offspring observable physical features is called a genotype. Phenotype is determined by the genotype and refers to the organism's appearance, characteristic, behavior, and development, or physically observable features. The number of time of trait appearance in the offspring after crossing the genes or alleles of the specific trait identified through the genotypic ratio. The genotypic ratio is better understood through the Punnett square. Punnett square shows all the possible traits or genotypes of the new offspring in graphical or table form after the crossing of homozygotes.
Although we commonly refer to different alleles as being dominant or recessive, dominance is a property of the phenotype. There are many forms of dominance: complete dominance, incomplete dominance, and codominance. Incomplete dominance, there is no difference between phenotype between homozygous and heterozygous individuals. In incomplete dominance, the phenotype in a heterozygous individual is visibly less intense than that in an individual homozygous for the dominant allele, so that homozygous and heterozygous genotypes produce different phenotypes. Hence, the heterozygous will have a phenotype intermediate between of a homozygous and a heterozygous individuals. Incomplete dominance is defined differently as follows. The incomplete dominance is referred to as the dilution of the dominant allele with respect to the recessive allele, resulting in a new heterozygous phenotype. For example, the pink color of flowers, such as snapdragons or 4 o'clock flowers, the shape of hairs and hand sizes, voice pitch in humans. The intermediate trait appearance in between the phenotypes of homozygous traits in the heterozygote is called incomplete dominance. the formation of a third phenotype specifically with traits and results from the combination of parent alleles is known as incomplete dominance, or the incomplete dominance is referred to as intermediate inheritance in terms of trait expression, and none of the alleles from the paired alleles expressed over the other for the specific trait. According to some definitions, there are several assumptions about incomplete dominance. An incomplete dominance occurs due to the combination of parent alleles, both dominant and recessive. Whereas several definitions define incomplete dominance as a phenomenon in which the heterozygote produced possesses an intermediate trait between the two homozygous traits. Moreover, some definitions from incomplete dominance in which the new offspring has a specific trait in less intensity than the dominant trait among the paired alleles. In other words, the trait is neither dominant or recessive.
The situation in which the phenotype of the heterozygote is clearly manifested is a cross between two homozygous phenotypes. After the combination of homozygous alleles or F1 generation, the heterozygote will have the intermediate trait. At F2 generation, it then shows a ratio of 1:1 phenotype in which the two are intermediate traits and other are either dominant or recessive traits.
Mechanisms of incomplete dominance. Mendel's experiment shows complete dominance after using the pea plant's traits, round and wrinkled, meaning the pea plants with specific traits, round and wrinkled peas, were crossed. This results in pea plants with round peas showing round as a dominant allele. Thus, the dominant allele was expressed over the recessive allele, that is wrinkled peas. Keeping Mendel's work under is wrinkled peas. Keeping Mendel's work under consideration, Karl Correns performed an experiment on 4 o'clock flowers. He took two true breeding flowers traits, red color as a dominant allele and white color as a recessive allele, of 4 o'clock flowers and crossed them. The results show an intermediate heterozygote with pink color flowers. None of the alleles get dominant. This situation in inheritance is known as incomplete dominance.
How does incomplete dominance work? To understand the mechanism of incomplete dominance, the botanists use Punnett square. The Punnett square predicts the genotype of the breeding experiment. In this case, one plant producing red flowers and another plant producing white flowers are crossed. The above Punnett square result in heterozygous offspring with an intermediate trait of pink color, showing that no allele gets dominated over the other. The two alleles are not expressed in a way to hide the effect of the other allele. Instead, the phenotype is in between the two and intermediate. Thus, the heterozygote is one that produces flowers with a pink color.
Incomplete dominance examples. Incomplete dominance is a widely studied phenomenon in genetics that leads to morphological and physiological variations. The pink flower color trait, which is an example of incomplete dominance, occurs in nature, such as those found in pink flower-bearing angiosperms. Turns out the dominant allele is not expressed completely, as shown in figures 1 and 2. Incomplete dominance, Punnett square. Apart from plants, incomplete dominance also occurs in animals and humans. For example, hair color, eye color, and skin color traits are determined by multiple alleles in humans. Take a look at the examples below for the complete dominance in plants, humans, and other animals.
In plants, the carnation of plant, which is an example of incomplete dominance, has true breeding white flowers and true breeding red flowers. A cross between white and red flowering carnation plants may result in offspring with a phenotype of pink flowers. 4 o'clock flowering plants are an example of incomplete dominance. Red and white flowering plants breed to produce offspring with pink color flowers.
What is multiple alleles? Multiple alleles are 3 or more alleles for a particular gene. Multiple allelism, biology definition, the condition of a presence of multiple alleles. An example of multiple alleles is the ABO blood group system in humans. Human gene I for isohemagglutinin, exists in 3 allelic forms: IA, IB, and IO. IA and IB produce type A and type B antigens, respectively, on the cell surface of RBCs, whereas IO or I is recessive allele and does not produce antigen. So, for instance, an offspring that got both IA and IB alleles will have blood type AB, and that who received IO would have blood type O.
Gregor Mendel suggested that each gene would have only 2 alleles. Alleles are described as a variant of a gene that exists in 2 or more forms. Each gene is inherited in 2 alleles, that is, one from each parent. Thus, this means there would also be having 2 different alleles for a trait. Offspring who describe different alleles of a gene are described as heterozygous, whereas those that have genotypes made of the same alleles, that is a gene for a particular trait, are described as a homozygous.
Although humans and all diploid organisms can have only 2 alleles for any given gene in genetics, multiple allele traits may exist at the population level. Thus, multiple alleles are important in promoting variation within the same species. In Mendelian inheritance, a given chromosomal locus was occupied by two distinct types of gene alternatives, one dominant and one recessive. These alternatives are two alleles of the same gene. However, in the given population, there are instances wherein there are more than two alternatives alleles existing. Those instances are referred as multiple allelism, where different forms of the same gene exist within the population. These three or more variants for the same gene are called multiple alleles.
When does multiple alleles occur? Multiple alleles exist in a population when there are many variations of a gene present. In organism with two copies of every gene, also known as diploid organisms, each organism has the ability to express two alleles at the same time. They can be the same allele, which is called a homozygous genotype.
Some example of multiple alleles: coat color of cats. For thousands of years, domestic cats have been bred to achieve a variety of coat colors. The gene that determines the coat color of the cat appears to have multiple variants, as the coats could range from black, orange, For thousands of years, domestic cats have been bred to achieve a variety of coat colors. The gene that determines the coat color of the cat appears to have multiple variants, as the coats could range from black, orange, brown, and etc. to white. This means that there are multiple alleles responsible for the coat color. The coat color gene has multiple alleles in the population, and the pigment producing protein will depend on the inheritance and the expression of these alleles. Other genes regulate curliness, shading, patterns, and even texture in a similar manner. The number of possible combinations and expressions of distinct genotypes from these alleles result in a wide variety of breeds, even when only 4 alleles are shared between two parents for each gene. The variation can be astounding.
Take a look at Figure 1. The coat color of cats clearly indicates multiple allelism, as it appears there are more than 2 alleles for the coat color phenotype. That is patches of black and orange or tortoiseshell pattern, black, gray, white, and patches of white fur or piebald spotting. Generally, the genotype of a cat can be determined by examining its coat coloration and patterns of coloration. If the phenotypes of the parental cats are known, it is usually possible to predict the coloration possibilities of kittens, though the calculations would be complex in most cases.
Multiple alleles among humans. There are traits in humans and other organisms that have 3 or more different types of alleles, genes. When a trait has 3 or more distinct alleles, we refer to it as having multiple alleles inheritance. The human ABO blood type alleles trait is an example of a trait with multiple alleles. 3 distinct alleles exist: allele A, IA, allele B, IB, and allele I, IO or I. If the allele A is present on the chromosome, protein A is produced, and the red blood cells of that individual contain protein A on their membrane. If the chromosome contains the allele B, protein B will be produced, and the red blood cells membranes will contain protein B. Finally, if allele I is present on the chromosome, If allele I is present on the chromosome, neither protein A nor protein B will be synthesized. These 3 alleles make up the ABO blood group trait. Figure 2. Possible blood types of offspring from a cross between male and female parent with blood type A and blood type B, respectively.
Thank you for staying with us until the end of our discussions.